Hello, everyone. All right, we're going to move on. We talked about the structure of DNA last week, and today we're going to talk about DNA replication. So if you remember, we have over 100 trillion cells in our body, and they're always making copies. Anytime you have damage to a cell, whether you get a little cut on your skin or your liver cell, remember in 2 years you have an entirely new liver. So we're always having to make copies of cells. And when we make a copy of a cell, we have to copy all the DNA. So DNA replication is what occurs anytime we have to make a new cell and we've got to get a new copy of DNA. And we call this process semi-conservative. And the reason it's called semi-conservative is because it uses the old DNA, which is demonstrated here, or what we would call the original strand. So this blue strand here and this blue strand here. And that uses a template to make a new strand. So when we make the new strand, or when we make a new copy of DNA, one is from the old strand. Remember, it's a double helix. And the other strand is from a new strand that was synthesized based on the old strand. Okay, so we call that again semi-conservative because each new DNA molecule has one of the two original DNA strands. Now, in order to understand and successfully perform a DNA replication, you have to understand the base pairing rules. Remember, that's the idea that A always bonds with T and G always bonds with C, or adenine always bonds with thymine, guanine always binds with cytosine. Okay, so that's how the old strand is a template. All right, so let's look at this. The green is the original strand, okay? It's pried apart, and we're going to talk about the key enzymes, and you're going to be held responsible for a couple of them. But it's pried apart, and then if you look, this green is the new strand, or the old strand, and the orange is representing the new strand. So here we have the green and the green, so these are the two original strands, and they were base paired, right? A with T, C with G, G with C here, A with T, T with A. When we pried them apart, now we're going to make a new copy of the complementary strand is what we would call it. So the T base, thymine, comes in. The G base, guanine, comes in because C and G go together. Now here we have G on the original strand, so it's going to bring cytosine, okay? And it goes on, and same happens here. This was the green on this side is the original, right? So now we're going to bring an A, bring a C, bring a G. Now remember, we always, when we start to talk about DNA, simplify it into the simple base pairing rules, which there's actually much more to this nucleotide than just their base, right? You have the 5-carbon sugar and the phosphate, which make up the backbone of the DNA. But the only way we can identify these nucleostrands as different are these bases. So now we have the two new DNA molecules, and if you see, half is the original, which is represented in green, and half is the new, which is represented by the orange, okay? And yes, this new looks identical to this original strand over here, okay? All right, if it were that simple, we could be done, but we need to know the key players in it, okay? So here's the first process. Each DNA strand becomes a template, okay? The proper base pairing rules, the base pairing rules become very important, the Chargaff base pairing rules, okay? When you watch other videos, they're gonna talk a lot about the direction of DNA replication. We're going to touch on it, but this is a hard concept that I don't want you to get too stuck in the weeds. What I'd like you to really focus on are the main enzymes, what they do in this process, and we'll talk a little bit about this replication direction in a minute. Oops, sorry guys. I touched the table and it knocked you out, but I think we're back. All right. So DNA replication takes place in two directions, but basically it's unwound and one end goes this way and one end goes this way. This is the thing I don't want you to get too stuck on, okay? Let's just look at these main enzymes and let's focus on them, okay? The main first enzyme is helicase, and helicase's job is to basically unwind these two